Okay, guys, and welcome back. In this video, we're going to be looking at the overall structure of the nephron as being the functional unit of the kidney. Now, something you're going to have to do again, as one of those skills that they might ask you to do in an exam, is to be able to draw and annotate a diagram of the nephron. Sorry, not to draw it, just to annotate it. Obviously, annotate means kind of label it and give a little explanation as to what it does. So let's look at some of the key parts that we've got to remember. You'll be familiar with this area over here. You've got the efferent arteriole coming into the glomerulus. So this is the glomerulus right in here. It's like a tight knot, high pressure capillary beds, and it's the site of blood filtration. Okay, so the filtrate then filters out into what we call the Bowman's capsule, okay, which is this area around here. It's a cup-shaped structure with a very, very porous inner wall which collects the fluid filtered out of the glomerulus. That feeds into our proximal convoluted tubule, which is here. It's a highly twisted section of our nephron with cells in the wall having many, many mitochondria and microvilli projecting into the lumen of the tube. So this is where remember 80% of what gets absorbed is reabsorbed. So the glucose, amino acids, sodium, and chloride ions. This is where it all gets reabsorbed. We then come down the proximal convoluted tubule into something called the loop of Henle. Now you can see there's a dividing line here, okay, and this is the dividing line between the cortex of the kidney and the medulla of the kidney. And so this is the what we call the descending loop, part of the loop of Henle. And on the other side we have the ascending line of the loop of Henle. Now this is basically where urine is concentrated, and where we'll come back to that. It's the next thing we're going to be looking at. We then have the distal convoluted tubule, which is another highly twisted section, but with fewer shorter microvilli and fewer mitochondria. We do have some reabsorption happening here as well, and we're going to come back to that again as well. And then finally we have our collecting duct, which is a wide tube that carries the filtrate back through the cortex medulla to the renal pelvis, okay, which is our concentrated urine, which is what we'll excrete. We should also mention the major blood vessels. We have the efferent blood vessel, the efferent arteriole, which is the one coming into the glomerulus. We have the afferent arteriole, which is the one that's coming out of the glomerulus here. We have the glomerulus, which is also, remember, a very specialized type of capillary network. We have the peritubular capillaries. Okay, now the peritubular capillaries can sort of be found in this area around here. They're a low pressure capillary bed that runs around the convoluted tubules, absorbing fluid from them. Okay, so it runs around both the proximal and the distal convoluted tubules. And then we have another blood vessel called the vasa recta, okay, which is an unbranched capillary that is similar in shape to the loop of Henle, and it kind of follows it around back up again. And then of course you have the venules, which carry blood to the renal vein, okay, which you can see here. Okay, so this will be coming back to our venule. So that's the overall structure and function. Of course, you have to practice drawing it, labeling it, and making sure we know it. Okay, so now we're going to look more closely at the loop of Henle itself. Okay, now we said we have a descending limb of the loop of Henle and an ascending limb of the loop of Henle, which goes back up again. And now some important features of this loop of Henle that are very, very special is that the descending loop is permeable to water, which means water can go out of it, but it's impermeable to sodium. Okay, so sodium, any sodium that's coming into it has to stay where it is. All right, so this is where the direction the filtrate is coming from. So water can move out across the membrane, but sodium ions cannot. Okay, on the opposite side, you have the ascending loop or the ascending limb, this is kind of the opposite to the descending, so it's impermeable to water but permeable to sodium. Okay, and there's actually sodium pumps edged along inside this membrane. So the overall effects at the moment is that the filtrate volume decreases, okay, because the water is going to be moving out of here by osmosis, and the output is slightly more dilute, and that over here there is going to be a large amount of salt being removed. Okay, so let's look at this a bit more closely. These red arrows here show that sodium ions are being pumped into the medulla, okay, by active transport. Remember, this section all around here is medulla, and kind of at the top here is where you'd encounter the cortex. So sodium is pumped into the medulla by active transport, setting up a sort of a charge differential. So chlorine ions are able then to diffuse out freely, following that gradient, okay, going from this negative to a positive area. So the solute concentration in the filtrate decreases as the loop ascends, okay? So as those sodium ions are being pumped out, the solute concentration in what's going up here is going to be decreasing, okay? And this is what we call a countercurrent effect, and I'm going to come back to this again in a second. So we've said the solute concentration in the filtrate decreases as the loop goes up, and so as the solute concentration increases here in the medulla, we've got an osmotic gradient being produced. Okay, and so as that filtrate comes down in here, water is going to move by osmosis into the medulla. Okay, so you've got water leaving here and sodium leaving over here. So the whole idea of the function of the loop of Henle is the recovery of water and salts from the urine. Okay, so remember, we need to keep water and salt in our bodies often. Okay, they're things that we don't necessarily want to leave, and so we want to sort of store it up inside the medulla. The longer the loop of Henle, it means the more water and salt that is able to be recovered. And again, we'll come back to that again in a little bit. And so the loop of Henle causes a countercurrent exchange. Okay, and what we mean by that, we say it's countercurrent because the fluids flow in opposite directions to one another, and it's a countercurrent multiplier because it causes a steeper gradient of solute concentration to develop in the medulla than would otherwise be possible with a concurrent system, as if the currents were flowing in the same direction. Okay, so you can see here this is coming from the proximal convoluted tubule, and these numbers here are giving you the sodium concentration, okay, in what we call milliosmoles, okay, and you might see this as MOSM. Osmoles are just simply a unit for measuring the osmolarity of something, of a solution. So you can see here you've got the solution has 300 osmoles coming in. Okay, now as that goes down, and as it runs down here, remember water is leaving, so this is going to become more concentrated. And then as it goes back up again, the sodium is actively being pumped out into here, which again raises the solute concentration. Okay, so it's going from a 100 to 1200. Okay, which again is creating a osmotic gradient. So as more water enters, more water is drawn out. Okay, and it just keeps going around like this, so that more water and more salt can be stored inside the medulla. Okay, so that we can keep it in our bodies. I think that's all we're going to talk about in this short video. Remember to check out some of the animations if you don't quite understand it, and to talk it through in class if you need any help.